Welcome back. In the last lecture, we talked about electrical activity in the brain, the fundamental currency of neurons' action potentials. Today, we'll talk about the chemistry of the brain, and what we'll focus on is neurotransmission, synaptic communication from neuron to neuron. Neurotransmission between neurons underlies everything your brain does, all the communication that your neurons engage in when they talk to one another, and so we're going to focus on that today. And furthermore, we'll use that information to talk about some of the drugs that affect brain function, drugs that emulate what neurotransmitters do. And these drugs are chemicals that come up in most people's lives, more than you might expect. So, as I said, neurons communicate by secreting chemicals called neurotransmitters. Now, this fact was an open question as recently as 50 years ago, and over 100 years ago, one of the pioneers of neuroscience, Camillo Golgi, had a different idea, the reticular theory, and his idea was that neurons communicated with one another by being joined to one another, by forming a continuous reticulated network such that there perhaps were not even individual cells. As it turns out, in the ensuing time, it's now agreed that his rival, Santiago Ramon y Cajal, formulated a doctrine, the neuron doctrine, which we now understand as being how neurons communicate, which is that they have little gaps between them, and those gaps are synapses, and those synapses are how neurons communicate with one another. Now, there is something that brains do that's similar to Golgi's old idea, and that's the idea of the electrical synapse. Electrical synapses have been around for as long as animals have been around, and indeed, electrical synapses are a major way that jellyfish use to form their coordinated contraction. And electrical synapses are an easy way to think about cell-to-cell communication, because what they demonstrate is the possibility that one neuron can talk to another one by shooting current into it. So electrical synapses are direct connections between neurons. And this is reminiscent of this old reticular hypothesis I told you about. And electrical synapses are how jellyfish nervous systems and how other primitive synapses actually work. But in fact, if you look in animals, including us, the most common means of communication is, in fact, chemical communication using neurotransmitters. Now, for this lecture, I want to convey to you the general concept that overarches this whole lecture, and it is that neurons release neurotransmitters, and those neurotransmitters act on receptors. And receptors are protein molecules that sit in the membrane of the cell, and they bind to neurotransmitter, and that binding causes a shape change in the receptor, and that change in receptor shape then is a fundamental step, a key step, underlying nearly all communication in the brain. And so this translation of electrical activity into chemical activity and then back to electrical activity is a recurring motif that happens all the time in your brain and in mine. Now, chemical synapses, as I said, are more common in most brains, both vertebrate and invertebrate, than electrical synapses. Chemical synapses have several advantages that direct electrical connections don't have. There are three advantages. One advantage is they are able to be inhibitory, so that one neuron can not only excite another neuron and cause it to be more likely to fire, neurotransmitter release and chemical neurotransmission allows one neuron to inhibit another neuron and keep it from firing. Another advantage is that neurons can communicate with one another in a directional fashion. So if you have an electrical connection between two neurons, then they can talk back and forth. But if you have a chemical synapse, then one neuron can release neurotransmitter to the other neuron, and that's directional signaling from the presynaptic side of the synapse to the postsynaptic side. So that's the second advantage. And the third advantage is that when receptors are used to receive messages from another neuron, then it becomes possible for those receptors to trigger biochemical signaling inside the neuron. And that biochemical signaling inside neurons is a critical step in processing information and also in guiding long-term processes such as learning and memory. Now, let's back up a little bit. I've asserted to you that neurotransmitters exist and they are how neurons talk to one another. But let's ask the question, how do we know that neurotransmitters exist in the first place? And for that, I want to describe to you a critical experiment in the history of neuroscience that proved this point. And it was an experiment done by the physiologist Otto Loewi. Now, Otto Loewi worked in the 1920s, and in 1920, he noticed that stimulation of the vagus nerve, so what he did is he was working with frog heart, and he found that when he stimulated a nerve that went to the frog heart, a nerve called the vagus nerve, when he stimulated that nerve electrically, what that led to was a slowdown of the frog's heart. So it would slow down and its contractions would get smaller. And so the frog's heart, which was able to beat in a chamber, just hanging there suspended in a chamber, could be slowed by his experimental manipulation. And the question is, well, how could this be? How could stimulation of the nerve lead to slowing of the heart? And the two predominating ideas at the time were that perhaps it was some direct electrical effect of the nerve acting to inject, say, electrical current directly into the heart, or perhaps some chemical messenger that was secreted and then picked up by the heart, say, through synapses, as we would say today. And he was puzzled about this, and he thought about how to do an experiment to test this. And he went to sleep, and he had a dream. He had a dream about how to do the experiment. And in his dream, it became clear to him how he would do the experiment. And he woke up, and he couldn't remember the dream. So this was bad because, you know, he knew it was a good idea, but he couldn't remember it. So another night, then, he went to sleep, and he had the dream again about exactly how he would test the idea of neurotransmission and chemical neurotransmission. And this time he woke up, and he wrote down his dream. And great, he went back to sleep. And then he woke up in the morning, and he could not read his handwriting. Okay, so that's still not good. Third time, he had the dream, and this time he woke up, and he went to the laboratory, and he did the experiment. And he did the experiment, and it worked. And this is the experiment that he did. So I told you that a frog heart suspended in solution in a chamber is able to beat and to slow down when activated by the vagus nerve. What Loewi did was he suspended 2 frog hearts in 2 chambers. And then he connected those chambers with one another by a tube, so that one frog heart is in one chamber, another is in another chamber, and there's a thin tube connecting the two so there could be communication between the two. And what he did then was he stimulated one heart's vagus nerve, and he then saw that its beating slowed down and got smaller, as I described to you before. But after some delay of a few minutes, what he then saw was that the other heart began to slow down, and its magnitude of heartbeat began to diminish. And so what that demonstrated was that something passing through the tube was responsible for the slowing of the heartbeat. And Loewi called that substance, he was German, he called that communication chemical Vagostoff, for stuff coming out of the vagus nerve. And we now know this chemical is acetylcholine. And it turns out that acetylcholine is a widespread neurotransmitter. And indeed, it is the neurotransmitter that is used at neuromuscular junctions. You recall that I showed you before a motor neuron innervating a muscle fiber. Acetylcholine is the transmitter that's used for a nerve fiber to talk to a muscle fiber to cause the muscle fiber to contract. Okay, so now that's the evidence that we have for neurotransmitter release. All right, now fast forward to the present day. What do we now know about neurotransmitters? Well, from Loewi's day, now we've found out a lot. And what we now know is that neurotransmitters are synthesized inside neurons. They're stored in axon terminals, the ends of axons where the synapses are formed with dendrites. And they're stored there until an action potential arrives. And when the action potential arrives, it arrives at the terminal and then causes secretion of neurotransmitter. And then the neurotransmitter is released and then diffuses to the other side. Now, any given neuron in the brain generally makes a very small number of neurotransmitters, most often only one. So for example, a neuron might secrete glutamate, which is the most widespread neurotransmitter in the vertebrate brain. Glutamate is an amino acid that can cause excitation. It can make its target neuron, the postsynaptic neuron, more likely to fire. And so glutamate is quite a widespread thing to find in neurons. However, a neuron cannot secrete both glutamate and another neurotransmitter, GABA, and GABA is the main inhibitory neurotransmitter of the brain. And the reason that a neuron can't make both glutamate and GABA is that there are enzymes that turn glutamate into GABA. And so as a result, if the enzyme is there, then a neuron is going to make GABA, but not glutamate. And if the enzyme is not there, if the neuron secretes glutamate, then it's not going to make GABA. Okay, and so there's a general principle that neurons make very small numbers of neurotransmitters. Now, it used to be thought that a neuron made exactly one neurotransmitter, so that every neuron only made one transmitter. And that is a belief that was widespread in neuroscience. It was formulated by Sir Henry Dale, and therefore is called Dale's principle. Now we know that there are exceptions. So even though Dale's principle is mostly true, it appears that there are exceptions that we have to consider when thinking about what individual neurons do. So for example, A neuron could make, say, a small neurotransmitter like one of these amino acid or amino acid-based transmitters I was talking about, glutamate or GABA, and it could also make a peptide. A peptide is a chain of amino acids that makes a somewhat larger neurotransmitter molecule. So one neuron could make a small neurotransmitter and release a peptide, and it could co-release those neurotransmitters. So there are exceptions to Dale's principle. But Dale's principle is still a generally useful principle to think about when we talk about a neuron being glutamatergic, that it secretes glutamate, or whether it's GABAergic, which means that it secretes GABA, or dopaminergic. All right, so these are ways that we think about neurons and the neuron types of the brain. Now, the major neurotransmitters of the central nervous system, the brain, are based on amino acids, as I've said. And the major neurotransmitters of the brain are glutamate, GABA, and glycine. And these are the main neurotransmitters for the vast majority of neurons in the brain. Nearly all the neurons of your brain secrete one of these three transmitters. Now, I said nearly, and I'll come back to that in a moment later on in this lecture, because the exceptions are in fact very important. Now that we've gotten to the point of talking about these neurotransmitters, now the next question is, how do neurotransmitters convey their message to another neuron? I've said that chemical synapses are gaps at which one neuron can talk to another neuron. That gap is about 20 to 40 nanometers, and so that's a very, very small gap. And it's about 6 to 10 times wider than the thickness of the membrane, which is itself about 3.5 nanometers thick. Okay, so these are very small gaps. And what happens is that neurotransmitter is released on the presynaptic side, diffuses across that tiny gap, and then binds to receptors on the other side of the synapse. And that's how neurotransmitters typically work. Now, what are receptors? The way to think about receptors is that receptors work according to a concept that was originated a long time ago called the lock and key mechanism. And the lock and key mechanism works as follows. Imagine that a receptor is like a lock, and the neurotransmitter molecule is like a key, and the key goes into the lock and is able to cause the lock to turn, and then you can get the lock to do something, and then you get into the house or whatever. So the general concept is that receptors, not only in the brain but throughout your body, are molecules that bind to keys and have a key that goes to them. So there might be a specific key that's good at opening that lock, and that specific key then turns the lock. And in the context of what we're talking about, the key would be glutamate or GABA or glycine. And then the lock itself is the receptor for glutamate or GABA or glycine, right? So we have glutamate receptors, GABA receptors, and glycine receptors. Now, these locks, what do they do? Well, at most synapses in the brain, these receptors are ion channels. So remember that I talked about sodium and potassium channels as being the basis of how action potentials are generated. Those ion channels were activated by voltage, and so they were channels that were activated by voltage and could be triggered by some kind of feedback mechanism to generate the action potential. Now, these ion channels that I'm talking about here, these glutamate receptors and GABA receptors, are now not activated by voltage, but instead they're activated by neurotransmitter. And so they are neurotransmitter gated channels, so they're like a gate that opens up to let ions through, and then the neurotransmitter shows up and binds and then causes the channel to open, and that's a neurotransmitter gated ion channel. And we often refer to the neurotransmitter as a ligand. Ligand is a technical term for the molecule that binds to the receptor. And this concept of a ligand is important because the ligand can be not only the neurotransmitter, but it can be other molecules as well, some of which, in fact, are drugs that can be taken to affect brain function. So the ligand concept is an important one in understanding how receptors work. Okay, so now we've defined what an ion channel is, what a neurotransmitter receptor is, and what a ligand-gated channel is. Now these channels open up, and then they're ion channels, and so they let ions into or out of the neuron, and that motion of ions then leads to a change in voltage in the cell. And that change in voltage is what leads to the chemical signal being turned into an electrical signal. So, for example, if the ion channel makes firing more likely, let's say it opens and lets positive charge into the neuron, then that increase in likelihood of the neuron firing means that that's an ion channel and that's a receptor that transmits an excitatory signal. And so typically, I said that glutamate is the most abundant neurotransmitter in the brain. Glutamate is typically an excitatory neurotransmitter. So neurons release glutamate, and glutamate opens ion channels, and then those ion channels let positive charge into the neuron, and that excites the neuron. Now, 90% of the neurons of the brain are glutamatergic, so most of the business of what our brains do is release and receive glutamate as a transmitter. This is a major, major thing that our brains do. And these excitatory neurons that secrete glutamate can project long distances. So for instance, when a neuron sends an axon to another part of the brain, or if a neuron sends an axon all the way down to the spinal cord, those neurons are glutamate-secreting neurons. Those are glutamatergic neurons, and something like 90% of the neurons of the brain are glutamatergic. So that's a major thing that brains do. Now, I should say as an aside that, that glutamate is also of interest, and we will come to this when we talk about taste and smell. Glutamate is also of interest because it's used in our diets. It's used on food to make food taste better. And it turns out that there is some concern about glutamate in the form of MSG giving headaches and causing bad health reactions. I want to point out that our brains have grams and grams of glutamate. Each one of our neurons secretes buckets of glutamate, and our heads are basically filled with glutamate. And so the amount of glutamate you would ever eat when you go out to get, I don't know, Chinese food or what have you, would be a very small amount of glutamate compared with the total amount of glutamate in the brain. And the brain has very good mechanisms for dealing with glutamate. So in fact, in MSG, it's probably the S, the sodium, that's probably giving people headaches as opposed to the glutamate. Okay, enough about glutamate. Let's now talk about the remaining 10% of the neurons. Most of the remaining 10% of neurons are inhibitory. And inhibitory neurons make connections that release typically GABA or glycine, and GABA and glycine are released by the axon terminals, and then those neurotransmitters then again bind to ion channels. And inhibitory neurons typically are local. They typically don't project very far. They just send local connections over no more than about 1 millimeter or so. And those inhibitory neurons, therefore, are local neurons that are often called interneurons. And that's a minor neuron type of the brain, but it plays an important function in making their targets less likely to fire. And so those are inhibitory interneurons. And the way that inhibitory interneurons work is by letting positive charge out, or perhaps by letting negative charge in. And in both cases, what that does is it makes the neuron less likely to fire. Another thing that those neurotransmitters do is that just by opening channels, they make the membrane a little bit more leaky, so it's harder to get the membrane to change in its voltage. And in all these cases, the action of these neurotransmitters is inhibitory and makes the neuron less likely to fire. Okay, now that I've talked to you about excitatory and inhibitory synapses, let's think about how they all fit together. So imagine a cubic centimeter of brain tissue. A cubic centimeter is about as much volume as fits in a thimble. And this thimbleful contains all the things that I've been talking about: neurons, axons, dendrites, and synapses. 1 cubic centimeter of tissue contains about 50 million neurons. It contains several hundred miles of axons and several hundred miles of dendrites as well. So it's packed with axons and dendrites. And in the same cubic centimeter of tissue, you have 1 trillion synaptic connections. So these synapses that I've been talking about are very small, and there are 1 trillion of them in a thimbleful of tissue. 1 trillion is 1 million million. Okay, so all these connections I've been talking about are quite small and extremely densely packed in neural tissue. And if we look at this piece of neocortex, any given piece of neocortex is filled with neurons that are excitatory mostly, and some inhibitory neurons. And the excitatory neurons are approximately shaped like little pyramids. They're called pyramidal neurons, and they send their dendrites outward towards the external edge of the brain. And then they send an axon out to some other part of the brain, and they also send axons internally within that bit of brain tissue. And those pyramidal neurons are excitatory. They send excitatory projections, and they secrete glutamate. And that can happen both locally and also over long distances. Now, furthermore, in this tissue, there are inhibitory neurons, and this is a smaller number, as I said. And these inhibitory neurons are local to the tissue and just send projections locally within that part of neocortex, and they secrete inhibitory neurotransmitters such as GABA and glycine. And what they do is they project locally to neurons in their vicinity, both neurons of their own type, but mostly to pyramidal cells, and form inhibition. So this is the way that neural tissue is organized for the most part throughout the brain. Excitatory neurons that can synapse over long distances and also locally, and then inhibitory neurons that send local connections. So that's how all these parts fit together that I've been telling you about. Now, I told you that 90% of the neurons are excitatory, 10%, almost 10% are inhibitory. What I haven't told you about is the remaining very tiny fraction of neurons. There's a small number of neurons that are not in either of these two large categories. They're tiny in number, but they are very important, and I'm going to come back to them just in a little bit. The subpopulation of neurons is responsible for a lot, and towards the end of the lecture we'll get to that. But first I want to talk about general principles of how synapses in general fit into brain energetics and function. And I specifically want to talk about the presynaptic terminal. Now, I've told you that neurotransmitters are released from presynaptic terminals when action potentials arrive. And these presynaptic terminals dump out little packets of neurotransmitter into the synapse. Now, it turns out, oddly, that presynaptic terminals are not very reliable machines. In the brain, synapses are comparatively weak and unreliable. And here's what I mean by that. When an action potential arrives at a presynaptic terminal, goes down the axon, gets to the presynaptic terminal, usually what happens at that point is nothing. You might expect from what I've told you before that what usually would happen would be a little bit of a neurotransmitter is secreted. But in fact, most of the time at synapses where people have looked very hard, most of the time nothing happens. So most of the time what happens is an action potential arrives, and typically anywhere between 1 and 4 times out of 5, nothing is released. And that is odd. That's a mystery in neuroscience, and it's not entirely understood why that is, except that evidently these synapses seem to be made very small, small to the point of being unreliable, and something about that appears to be good for neural function. It seems to be that there's something optimal about having a very unreliable connection and packing, as I said, 1 trillion of them into a thimbleful of tissue. And there's something about that general plan that uses the tissue efficiently and well. So that's an odd fact in neuroscience that people are a little bit puzzled about. Now, there are exceptions to this. Some synapses in the brain and nervous system are quite reliable. So for example, at the neuromuscular junction, which we've talked about, that controls muscle contraction, those synapses are very reliable. And that makes sense because in fact you don't want your muscles to not do anything most of the time. You in fact want to be able to get away from danger and do something reliable. And so neuromuscular synapses are composed of many, many terminals, and those many, many terminals then can secrete dozens or even hundreds of vesicles. And these complex, many component synapses are much more reliable. And what that leads to is the fact that muscle contraction is quite reliable. And so a presynaptic action potential always triggers a muscle fiber to contract. Okay, so that's an exception to this, this unreliable synapse rule. Another exception is the squid giant axon, which is this well-studied axon that's been studied for many years, where many of these principles were first worked out. It turns out the squid giant axon, again, it makes a synapse that's very reliable, and that's part of an escape reflex that the squid has that allows it to get away from danger easily and reliably. So these are exceptions, but generally presynaptic terminals are in fact fairly unreliable devices. Now, the other thing is that single synapses are weak, and an individual synapse is not nearly strong enough. It doesn't give nearly strong enough of an electrical kick to cause the postsynaptic neuron to fire. And what that means is that typically around 100 presynaptic neurons need to fire at about the same time in order to get a postsynaptic neuron to fire an action potential. And so as a result, it takes lots of neurons ganging up at about the same time to cause a postsynaptic neuron to fire an action potential. Okay, so now, there's a second note about synaptic function, and I want to take all these things that we've talked about and connect them back to something we talked about before, which is brain energy use and functional MRI. Now, remember I said that the fMRI signal is triggered by neural activity, and I said that you should think about the fMRI signal as the outcome of a call signal in which neural activity sends out a message, I think we need more blood flow here. And the general biological principle appears to be that neural activity leads to blood flow. Now, and that blood flow could have some downstream effects like making more energy available for future needs. So, what is that call signal? What's the thing that's sent out by the neural tissue that says, please send more blood over here? Now, there are 2 major categories of activity that it could be. One major category is action potentials, these impulses of electrical activity. Another category is what we've been talking about, which is neurotransmitter receptor interactions. Now, as it turns out, the transmission of synaptic information costs a lot of energy. 3 scientists working in Germany, Alle, Roth, and Geiger, did a calculation based on what's known about how synapses work to figure out how much charge moves across the membrane, how many ions move across the membrane, both during the generation of a spike and also during synaptic transmission. And what they showed is that action potentials might be a pretty small part of the energetic budget. So despite the fact that they do use up energy, it turns out that synaptic processing, all this moving of charge through glutamate, glycine, and GABA receptors, potentially sucks up quite a lot of energy, perhaps the lion's share of brain energy. And so their calculation suggests that. Now there's a second line of evidence. The second line of evidence comes from doing local field potential recording. And this is a kind of recording that's related to EEG, which we've talked about before. And local recording suggests the same thing, that synaptic signaling events lead to increased blood flow. And so there is some evidence that from direct experimental measurement. So we have two lines of evidence. One is computer-based calculation, computer simulations, and the other, the direct measurement of brain activity. And these two kinds of measurements have the same implication. They imply that synaptic signaling costs a lot of energy. And so when we look at fMRI signals, what we should be thinking about is the possibility that what they probably reflect is synaptic signaling. They probably are reflecting local synaptic processing. So to summarize, the way to think about synaptic activity in terms of whole brain function is that individual synapses are not terribly reliable, but in the aggregate, they consume a lot of the brain's energy and may be a key event underlying the fMRI signal. All right, now let's continue across the synapse. We started with neurotransmitters, we've crossed the synapse, we've talked about the synapse themselves, the synapse itself. Now let's talk about the receptors. Now I've already described some receptors. The main receptors in the brain for processing many electrical signals are receptors for glutamate, GABA, and glycine. And these are receptors that are ion channels. There's another kind of receptor that's not an ion channel, and this other kind of receptor is a major type of receptor that plays a critical role in receiving signals from a vast array of other neurotransmitters. This other category of receptor is known as a metabotropic receptor. Metabotropic meaning that the receptor drives metabolism. So instead of letting ions in or out, what these receptors do is trigger the formation of chemical signals inside neurons, and therefore they move metabolism, so they're therefore called metabotropic. It turns out that the vast array of other neurotransmitters in the brain, other than glutamate, GABA, and glycine, largely use these metabotropic receptors. And these metabotropic receptors receive neurotransmitters from molecules that you've heard of. For instance, there are the biogenic amines. Biogenic amines are small molecules that will come up again when we talk about attention, emotions, and mood. And those molecules include dopamine and norepinephrine and serotonin. Other small molecules that mostly work through metabotropic receptors are histamine, peptides, which, as I said before, are small chains of amino acids. And it turns out that acetylcholine works on both ion channels and on metabotropic receptors. Now, all of these neurotransmitters are made by synthetic pathways. And again, Dale's principle shows up. And each one of these neurotransmitters requires particular enzymes, and so therefore an individual neuron usually makes one type of neurotransmitter, sometimes two if one of them is a peptide. Now, despite the fact that we've heard of these neurotransmitters, that they show up a lot when we hear about drugs and hear about brain phenomena, each of these minority neurotransmitters is only made by a very small number of neurons. So for example, of the 50 to 100 billion neurons in the human brain, only 200,000 secrete dopamine, so that's a tiny fraction. Yet dopamine plays a critical role in brain function in a wide variety of roles. And dopamine neurons, dopaminergic neurons, then send their axons throughout the brain. So despite the fact that these neurons are very few, their influence is quite widespread. And if we think about what all these minority transmitters are doing, think of the brain as having many different kinds of keys. Each one of these keys opens locks. And there are many kinds of locks. And in fact, each neurotransmitter opens more than one kind of lock. And so you can have multiple types of receptor that are all activated by one key. So one key can open multiple locks. And this is going to be important for the next lecture, because what we're going to be talking about is ways in which synthetically or in nature there are chemicals that can open these locks that are different from the native neurotransmitters. So the native neurotransmitters are agonists, and so what that means is that they are molecules that can agonize, that can turn on the lock, that can turn on the receptor. And it turns out that in nature, there are chemicals that can emulate the action of neurotransmitters, and those are also called agonists. There are other drugs, other chemicals found in nature that block the receptor, that prevent it from being activated, and those are called antagonists. And so two major kinds of molecules you can find in nature are agonists and antagonists, and these are ways that the receptor can be blocked. And furthermore, there are chemicals that can modify how the native neurotransmitter works. So there are other molecules that, say, can block the uptake of neurotransmitters. So neurotransmitters need to be vacuumed up after they're released, and that reuptake of neurotransmitter can be blocked. And that reuptake process is another way that drugs can act upon the brain. And in all cases, what's going on here is that these neurotransmitters are activating chemical signals inside neurons, and that action of those downstream chemicals then underlies a large part of what these minority neurotransmitters are doing. So that sets the stage for understanding how neurotransmitters work and how drugs, in fact, work on the brain. And that's going to be a theme that we're going to be coming to again and again in this course, the idea that external chemicals that we take, that we've synthesized, that we find valuable, are part of brain function. Okay, so now let's summarize what we've learned today. What we've learned today is about neurotransmitter release and about how chemical synapses work. And what we've learned is that neurotransmitters act on receptors, and they do so by jumping the gap at synapses, these tiny gaps that connect neurons to one another. We've learned that receptors are protein molecules that are usually found in the postsynaptic neuron's membrane, and when those protein molecules change shape, that change in receptor shape then leads to transmission of the signal. And that change of receptor shape can lead to either flows of ions across the membrane or chemical signaling steps inside the neuron, in the case of metabotropic receptors. And communication between neurons is founded upon receptor action, either ionotropic or metabotropic. Furthermore, we've learned that synaptic communication consumes a large fraction of the brain's energy, and this is a major component of what brains do. And finally, we use chemicals to purposely affect these receptors and therefore influence brain function, and those are chemicals that we call drugs. And what we'll do in the next lecture is talk more about how people affect brain function with drugs, either legally or illegally.